So this is going to be a slightly a bit more complicated video, so pardon my drawing, it's really horrible, but let's talk about first about the flow cytometry. First of all, what is the flow cytometry, right? So it's a technique that we use in clinical laboratories to count cells. Basically, we can count the cells in your peripheral blood, we can count the cells in your bone marrow, and we can sort them out by size and complexity, basically. So we can tell you how many lymphocytes you have, how many monocytes, how many neutrophils, and things like that. So what is the basic principles here? This is the fluid dynamics. So we load our specimens on top right here, which is, let's say, it's a peripheral blood, and then this blood is going to be moved down to a different filters in such a way that in the injector site the cells will go one by one. They will be just shoot out one cell at a time. So as they travel, they will hit this laser, and based on the size and complexity, the light that is emitted from this laser is going to scatter, right? So one way to capture that light is called the forward scatter. So in this particular way, we'll have here a special filter that will block the central light. So we're going to measure only the scatter, okay? This is called the forward scatter. Now, based on the cell size, this scatter will be either too broad or too narrow, and basically it will be converted to some voltage, something like this, okay? So the thing is that majority of the cells are different not only by size, they are also different by complexity, by granularity, by cellular organelles and things like that. So this laser will have also a side scatter, okay? The light will scatter everywhere, but we can capture some of the side scatter and split it in different channels like that, okay? And convert it to a voltage and combine the forward and the side scatter both together into a single graph, and the graph will have the forward on the base versus the size. So every dot on this graph now will represent a cellular event. So the one which are light here, these are small size, non-complex, these are most likely to be lymphocytes. Then above it, okay, slightly more bigger in size and slightly more complex cells, these are the monocytes. And right on the top will stay neutrophils, because they have lots of granularity, lots of complexity, and much bigger in size than a lymphocyte. Now, okay, I think that's about it.